of course, the organizer of this workshop, and also thanking for inviting me. I feel a bit, must admit, I feel a bit on the edge of this workshop because I'm not going to present any models. I don't do much modeling myself, but I'm going to present empirical data. And also I'm going to be talking about plants. And basically I'm really interested in the coevolution between plants and humans, and also between crops and their wild relatives, because my main interest is really in plant domestication. And my sort of favorite model organism is maize. And so this is a picture here of a mural in Mexico City, where you can see Native Americans cultivating corn. So maize was domesticated in Mexico around 9,000 years ago. And today I'm gonna talk about the relation between maize and its closest wild relative. It's what we consider its ancestor, that is called the teosinte. And this study was actually initiated by Valérie Lecor, the team of Valérie Lecor at the UMR Agroécologie at INRAE Dijon with Mathieu Siol and Christophe Dely. And basically, Valérie Lecor and her team contacted us, so Yves Gourou at IRD Montpellier and myself at GQ Le Moulon, because they had discovered and also sampled a number of teosinte seeds in France. Basically, what we showed in this study is sort of summarized in the title, is that we found that adaptive introgression from maize has facilitated the establishment of teosinte as a noxious weed in Europe. So just maybe as oops, sorry, I'm trying to go to the next slide. I can't, oops. Sorry, I'm sorry, but yeah. There we go. Yeah, okay, sorry. Let's see. Okay. So this is just a slide to remind you a bit how we see the evolution of crops. This is illustrated in maize. So basically, what we usually considered is that everything starts from wild population that are found in natural environments. So here is a picture of a field of teosintes growing in Mexico. So teosintes are endemic to Mexico. And then from this wild population, early farmers have domesticated plants. So during the domestication process that occurred for most of plants during the Neolithic revolution. And this gave rise in maize to maize land races. So from teosинtes, you go to maize land races. These land races have been cultivated from millennia. And this cultivation is accompanied by both breeding and dispersal. So from its center of origin, maize was rapidly diffused all over the Americas. And more recently, with the rise of modern agricultures, these land races were to derived elite lines that are at the basis of the maize hybrids that you can see in most of our fields. This later stage of modern agriculture is also accompanied by breeding and dispersal. So what do we know about teosинtes, about annual teosинtes? So we know that they are endemic of Mexico, and we also know that there are two subspecies of teosинtes. One that is called Parviglumis. So some years ago, we modeled the niches of these two subspecies of teosинtes. So the one that is called Parviglumis, its niche is represented here in green. It's a lowland, it's a subspecies that grows in the lowlands at an average altitude of around 1,000 meters. It's also known to be the direct ancestor of maize. And then there is another subspecies that is called Mexicana, whose niche is described here in red, and that grows at an altitude of around 2,000 meters. So it's a subspecies that is adapted to the highlands of Mexico. And you see that those two subspecies have really distinct niches. So here are some picture of them. And we also found that these two subspecies are quite divergent. So using SNPs here, we have found a divergence between Mexicana and Parviglumis, and you can also see that within its subspecies, you have a sort of a genetic substructuring. So in Mexicana, you have two genetic groups, and in Parviglumis, three genetic groups. Like a few years ago, what we did is we evaluated in the field 11 populations from these two subspecies, which represented over 2,600 plants, and we evaluated them in two common gardens in Mexico, gardens that were located at mid-altitude. And we basically measured 18 traits, and this is the result of a multivariate analysis that, as you can see, revealed some phenotypic differentiation between Parviglumis here on the left in green and Mexicana in red on the right. And we also found evidence for an altitudinal syndrome. So here the colors, the lighter colors stands for lower altitude and the darker colors for higher altitude. And there is a trend from lighter to darker colors between the two subspecies along the axis 1 of the PCA. This is actually more easily observable, where instead of running a multivariate analysis, you do a trait by trait analysis. So we rank here the population by altitude from the lower to the higher altitude. You have the two subspecies, Parviglumis in green, Mexicana in red. And what we have done is that for each of these traits, so for each of the 18 traits independently, we have run some QST, FST type of analysis to see whether the phenotypic differentiation was greater than that expected from the neutral genetic differentiation. And we indeed show that among 18 traits, 10 are evolving under spatially varying selection. This is the case, for instance, for female flowering time, male flowering time, grain weight, all the traits that I am presenting here. And so we define based on this an altitudinal syndrome where plants growing at higher altitude, they tend to flower earlier, to have also larger and heavier grain, less stomata density and less tillers. So they are sort of less bushy phenotype. Okay, so I talked to you about the teosintes that occur in Mexico. I told you domestication occurred. So the oldest archaeological record dates back 8,700 years ago. So it was domesticated here, and then it expanded through the Americas. So you have different dates representing different archaeological records, and it was only introduced quite recently in Europe at the time of the discovery of the American continent. So the different colors that you can see in this map are different maize genetic groups. That's not so important here. Okay, what's important is that maize was is originally a tropical plant, right? Domesticated from teosinte in Mexico. And of course, such an expansion through the Americas has required major flowering time adjustments. So typically, if you take a maize growing in northern USA, so in the y-axis here, you have the flowering time, you see that maize growing in the northern USA flowers much earlier than the one growing in the southern USA, and in the middle USA, you have maize that flowers at an intermediate timing between those two. And people, of course, have searched for the determinants of this adaptation when maize moved towards northern latitudes, which required selection for early flowering under long days, because in the tropic, the days are short at the time of flowering. But moving in northern latitudes, you need early flowering under long days to adjust the life cycle. And people have shown that there are a number of genes involved in that adaptation, including one that is really a major player of photoperiod sensitivity that is called ZCN8. So you see that at ZCN8 gene, there is, for instance, this allele whose frequency increase as we move toward northern latitudes. Now, back to teosinte, what we also know is that the subspecies mexicana, so the one that grows in the highlands and which is not the direct ancestor of maize, is known as a weed that invade maize fields. So this picture was reported in 1921. And in this paper, the question was already asked whether wild teosinte can hybridize with cultivated maize. And the answer given was yes, it happens that sometimes this Mexicana teosinte do hybridize with maize. And also that they are weeds. So what are the weeds features? Weeds have direct consequences on crop production. They usually rapidly spray, and they display an agricultural weed syndrome. So usually they display seed dormancy, short life cycle, and high fecundity. Weed typically falls within two categories. Either they are unrelated to crops or they are related to crops. And crop-related weeds are usually the most problematic. They are very difficult to control because of the close genetic proximity with the crop. Teosinte in Europe is a very recent invasion. In fact, the presence of teosinte weeds was officially reported in 2016 in Spain and in France. In Spain, teosintes cause important yield loss, and they also do so in France, where they infest maize and soybean fields. According to a technical report, French teosintes were first observed in the early 90s and were only observed in 2010 in Spain. So it's a very, very recent invasion. The motivation of the present study was to investigate the establishment of this extremely recent weed. And more specifically, we wanted to elucidate the origin of the European teosintes, assess the genetic similarity between French and Spanish teosintes, and also identify the adaptive changes that have sustained habitat shifts. So what we did is we collected seeds from 8 maize cultivated fields in Nouvelle-Aquitaine. So that's Valérie Lecor and her team. And then we genotyped those teosintes, and we retrieved over 24,000 SNPs. So that's a very small amount compared to the size of the maize genome. But nevertheless, we recovered over 24,000 SNPs in a sample comprising over 1,000 individuals. These individuals were French teosintes, but also a compilation of data previously published, some Spanish teosintes, some Mexican teosintes from the subspecies Parviglumis, but also the subspecies Mexicana, and data from maize land races and maize inbred lines from the US and Europe. So first, what we did is using the SNPs, we performed a PCA analysis. And what we found is that both the French teosintes in orange and the Spanish teosintes in pink are actually closer to Mexicana, so the highland teosinte, than Parviglumis, the lowland teosinte that gave rise to maize. Maize is represented here in green and blue. So clearly Spanish and French teosintes are closer to Mexicana. This is also obvious in this structure plot, where you can see maize forming a blue genetic group, Mexicana the yellow genetic group, Parviglumis, a red genetic group, and you see that the French and Spanish teosintes are clearly from Mexicana ancestry, but they also display some introgression from maize, so some blue color here. Okay, so we run a trimix analysis to look at the history of split and admixture. And what we found, so we confirmed that both the Spanish and French teosintes are closer to Mexicana. They are actually, among Mexicana, they are closer to Mex1, which is the race Chalco that I talked to you about, happens to be a weed in Mexico. And the second thing that we found is that we found evidence of gene flow from dent maize to these European teosinte, both French and Spanish. We actually measured or estimated this gene flow and found that it was about 12% for French teosinte and 42% for Spanish teosinte. Then we run an assay to look at flowering time. So the team of Valérie Lecor planted some French teosintes in Dijon. They found that the French teosintes flower much earlier than the Mexican teosintes. The French teosintes actually flower at the same time as maize flowers in Dijon. But the Mexican teosinte, well, the Parviglumis, the lowland one, almost, for most of them, they did not flower at all. The Mexicana did flower, but very late in the season, so that they were not able to produce seeds, and they flower at a time where we typically harvest maize. We further look at adaptive footprints in European teosintes. So in order to do so, we used PC adapt. We used the square loadings of this first PC here to detect outliers among the 24 something SNPs that we genotyped. You see that this first axis differentiate quite well the Mexicana group from the French and Spanish teosintes, and we found a number of outliers, most of which actually fell along the chromosome 8. There is a cluster here of outliers along the chromosome 8. So I am presenting here the 10 chromosomes of maize, and I represent the outliers along the chromosomes as the red arrow, well, red cross here. So you see this cluster of outliers on chromosome 8. And what is shown here in the y-axis is the patterns of local ancestry in European teosintes as inferred by Eley. So these plots, so we basically consider here two source population, dent maize and the Mexicana teosintes. The race Chalco, and the plot shows the mean ancestry allele dosage of dense cultivated maize along the different chromosomes for the French teosintes in blue and the Spanish teosintes in green. So when you have an allele dosage of 2, it means that in this particular region of the chromosome, the ancestry, basically the French or the Spanish teosintes have 2 alleles from the dent maize. So if we zoom in that region of chromosome 8, where you have a cluster of outliers, what we found is that this region encompassed the gene ZCN8 that I mentioned before, that is a major player of maize adaptation to northern latitudes. And you see that in this region, there's also a very strong signal of introgression from the dent maize in European teosintes. This is true both for the French and the Spanish teosintes. Now we looked at another region located on chromosome 2 around the gene ACC1. So it's important to know that there is a non-modified genetic herbicide tolerant maize variety that is used in France since 2001. Herbicide tolerance is caused by a mutation at this gene, ACC1. And what we found here is that, well, we typed this mutation in our French teosinte, and we classified the teosinte as either homozygous for the resistant allele, RR, or sensitive, homozygote sensitive, or heterozygotes. We also apply some herbicide to the teosinte. We found that 43% of French teosinte are resistant, and all of them were from the RR genotypes. And in this region, we found that the dosage of introgressed allele from maize is actually consistent with the genotype at ACC1. So clearly, there is a genomic region around ACC1 that was introgressed from maize in French teosinte, and that confers resistance to this herbicide. We went further and sequenced basically this gene in French teosinte, and we found that the haplotype for the resistant teosinte, so genotype RR, is exactly the same as the one found in maize varieties resistant to this herbicide, while the sensitive allele was closer to the Mexicana allele. So clearly, this is again proving that this allele was inherited from maize. So in conclusion, we wanted to elucidate the origin of European teosinte. We found that these teosinte come from Mexicana race Chalco, a teosinte weed adapted to highlands in Mexico. Then we found that French and Spanish teosinte are differentiated. French teosinte were introduced earlier, yet they actually display less introgression from maize. We think that this may be due to the fact that there has been maybe more time for purifying selection to act against maladaptive allele in French teosinte, and maybe this purifying selection acts to also keep the weedy phenotype. And concerning habitat shift, we found that local maize has contributed to the adaptation of European teosinte. ZCN8 is likely a major contributor to the shift in flowering time for European teosinte, and ACC1 confers herbicide resistance in French teosinte. So I think the take-home message here is that introgression is an important source of adaptive variation. There has been in the past some examples where wild population contributes to the local adaptation of the crop. And I think here we illustrated one of the few examples where the crop can actually in reverse contribute to the local adaptation of a wild population. So here is really an example of crop to wild gene flow contributing to a niche shift and to the establishment of a weed. So introgression is important to consider when forecasting invasiveness risks. And again, I want to end this talk by thanking Valérie Lecor, Mathieu Siol, and Christophe Dely for offering us to collaborate in this project. And thank you for your attention.
Thank you, Maud, for this great